Well, now that I've done my controls, I am ready to do a trial run of my experiment. Make sure that I understand the setup well, that I know how to record my results, assess my results, know how long the reaction takes, and what it looks like when it's completed. So what I've got over here is 2 test tubes that are labeled S for salivary amylase and C for commercial amylase. I have timers, and I have a spot well. So first thing to do is to put iodine in all the wells of my spot plate, just 100 microliters of iodine in here. So when I transfer samples from my reaction, I can tell right away if the reaction is complete or not. So I will load them up. I might not need them all, but it's such a small amount of iodine that it's easier to have too much in there, too many wells filled with iodine, then needs to put more during the reaction. Now that I've got the iodine already, I've got my timers, I have my enzyme, I'm ready to start the reaction. So I will start with my salivary amylase over here. So in my test tube, I take a 1000 microliter micropipette, which is 1 milliliter. I will pipette 1 milliliter of my saliva sample, which is mixed, I have to say, with deionized water. That's a lot of saliva otherwise. And S for salivary amylase, this is the one I'll start to. As soon as I add my enzyme to the starch, I pre-measured 4 milliliters of starch, I will start the timer, mix the reaction, and start taking samples and see if the starch is breaking down with my salivary amylase. So I am ready to start, and here it is. So I will mix my sample and start taking 100 microliters sample. So we can see that this is still quite dark over here, so there's still lots of starch in this one. So I can wait a little bit before I take another sample. Now, since this is not happening very fast, I might as well run the second reaction at the same time. So I will do the same thing with the second experiment, run it at the same time, take the proper pipette, the C for commercial amylase, pipette 1000 microliters of my sample, and I have a second stopwatch for this one, so I will start my second stopwatch as I add the amylase. And here we go. I mix my sample, and it doesn't quite matter which well I test my sample in, because I'll see right away if it's working or not. So we are still a little ways from having completed results. I can take a sample of the first one again. Oh, I can tell it is digesting, but it's not done. So I will wait a little bit. Maybe take a sample of the commercial amylase, which is also digesting, a little bit behind our first sample, but of course it hasn't been running for as long. Go back to the saliva or salivary amylase sample. Still not quite complete, but getting there. As the color gets lighter and lighter, it's probably a good idea to take more frequent samples so that you know with more accuracy the time it really took for the starch to be digested in the conditions you're testing. When you're taking your samples, you should make sure that they are consistent, which means that what you call the reaction complete is having the same shade of yellow in the final well of all your reactions. That means it doesn't have to be bright canary yellow necessarily, but if it's curry colored or mustard color, as long as you call it done with all the reactions at all the temperatures or all the pH with the same shade of yellow, it's all right. We're testing which conditions are best for each amylase to digest starch, not necessarily which condition allows it to digest the very last little bit of starch in there. Once you have 2 or 3 wells of the same shade, we can call it done. So I will test this one again here, my salivary amylase. It was getting really close. I'm tempted to call this one done. It's definitely not blue anymore, not brown. And let's see what happened there with our commercial amylase. This one is still a little bit darker, so I would say right now the salivary amylase I will call done at 3.5 minutes. Exactly. And I'll wait a little bit for the commercial amylase. I'll take another sample and see how long it took it. A little bit more. Over here. And I'll do maybe one more sample. We can see that we have the same shade for several wells in a row. It's possible that the salivary amylase give it a more brown color when it's mostly done, as opposed to the more orangey color of the salivary amylase. But let's see over here. There we go. So I would say I'd be tempted to call it done. It is not bright canary yellow, but it's not changing much. And one thing for sure, it is not nearly as dark as it used to be in the beginning when it was dark, dark brown, almost black. So now that I've practiced my experiment, I'm ready to do my real trials with different pH and different temperatures. See if I've got the same or different results with the 2 different type of amylase. So far they've reacted very similar to each other in the amount of time it took them to digest the starch in our trial experiment.
Yeah, I stopped it. It's still going.